Hello everyone and welcome to our NGS-CN Summer School online collection on spatial transcriptomics. Before we jump to our next speaker, let's take a look once more who we are. So it's a next generation sequencing competence network that consists of 4 production sites as you see on the map. So in 2019, DFG has funded centralized sequencing applications. There were quite a lot of calls, and within each call there were a lot of projects. And from 2024, DFG has changed the strategy towards individual sequencing applications. That means that now there is no special deadline for a centralized call, and you can co-apply at the DFG together with the chosen genomic facility. If you wish, here is a QR code, contact us, and then we could take a look and maybe sequence together. If you want to know something more about our news and upcoming events, then you also can take a look at the QR code, scan it, and get into our mailing list. Let's grow community together. And with that to be said, let's welcome our next speaker.
Oh, thank you. Max gave you an introduction to into sequencing technologies. Then Susanne told you how you will prepare or how single-cell libraries are prepared and sequenced. And in the upcoming next talk, you will hear a bit about single-cell counts and how they are analyzed. However, there's one step in between that is missing and that is also important. And it is just the steps how you get from the reads to the counts. And today I will talk a bit about this step, explain you some basics about how to convert reads into counts, what is mapping, which pipeline you use, how the pipeline does the converting the data, and so on and so on. Please note, this is also, it's basic bioinformatics. So when you are new to single-cell data analysis, then this is a good introductory talk for you. My talk contains several parts, and in the first part, I just want to talk about single-cell data or raw data, sequence data in general, and how you could do like a first quality control. Then I will cover the topic how we get from reads to counts. And in the next part, I will then present a pipeline. It's called Cell Ranger. And this pipeline takes care of this step for you for a certain data type. I will come to this later. Finally, I will show you some of the output you can expect from the Cell Ranger pipeline and how to continue. So before I start here, I just wanted to give you, or Susanne already told you about single-cell technologies in general. And she explained it quite nicely. And therefore, I will just give you a quick recap of what is out there. For the analysis, many of the steps you will do for the analysis of these technologies are kind of similar. There are small details that change, but yes, okay. First of all, we have SmartSeq2, and SmartSeq2 is a plate-based technology, meaning now explained as a bioinformatician. You have here like a 96 well plate. You throw in each well a cell, you sequence, and you're done. It sounds very easy, right? This was probably the first single-cell sequencing technology. I think it's over 10 years old already. And you can do up to 1,000 cells because you have to sequence every well, and every well gets a data set. And then it quickly gets you end up at the end like 10,000s of files, which is not feasible anymore. And also it gets more expensive the more you do. Then there is probably the most widely used technology. It's called 10x genomics. I think it has been around for 8 years or something. And here cells are captured in oil droplets. These oil droplets have beads, and these beads are barcoded. And by this, we can identify the cells. 10x genomics is, you can use it for up to 10,000 or even 100,000 cells. and it works quite well. Furthermore, we have now then another of these fluidics. So 10x is a fluidics-based method, and BD Rhapsody is an alternative. It's also fluidics, but instead that the cells are encapsulated in this oil droplets, they sink into wells. So this is like a chip or a blade, and you have small, I think, nano wells probably, and the cells come from the top and slowly sink into the cells. And ideally, you want to have one cell per well, and then you can do the prep and the sequencing. BD Rhapsody is, as I said, already an alternative, and it is supposed to be good for cells that are sensitive. So because for 10x genomics, you have this fluidics method where you just have pressure based, so where the oil flows in one direction and the cells in another, and there's pressure applied, and this can be harmful for cells. And for Rhapsody, it's just cells just sink into the wells, and this is better for sensitive cells. And then we have two more recent technologies. There's Parse Biosciences, and this is a technology that uses combinatorial indexing. So basically what you do is you have cells in plates, you barcode them with one barcode, and then you do another round of barcoding, and I think in total 3 rounds of barcoding. So that in the end, you will have tagged each cell with a unique combination of 3 barcodes. This makes the method very scalable, so you can do small amounts of cells, but you can do also up to 1 million or more cells, which is quite impressive. And finally, we have PipSeq, and PipSeq is a very recent technology. It is developed, or was developed by Illumina, and here they use so-called partitions, chemical partitions to encapsulate cells. So of these technologies, in my talk, I will talk about 10x. As I said, many of the steps I will present you apply also to other technologies, but I chose this one to show you how data is processed. Let's assume you have done your single-cell experiment. You gave your samples to the facility or the sequencing center. They did library preparation and sequencing, and then they will send you raw single-cell data. This raw single-cell data is, so to say, these are the most unprocessed version of single-cell data you can think of. So it's your first step in the data processing. Typically, they will be sent out as files, for example, by email or by download. And often they are FASTQ files. For the FASTQ files, I should note that these raw FASTQ files, we need them for this step I will show you now, but later they are often not needed anymore. So you could think that, okay, now I have counts already. I don't need the FASTQ files. They are big, they are messy. I can get rid of them. But this is not true, and I would advise against it, because it could be that you need to reanalyze certain data, then you will need the FASTQ files. And also now every journal wants the raw data. So once you have done your single-cell data analysis, you have analyzed the data for months and have a good result, a nice UMAP, and so on and so on. But then the journal wants to have the FASTQ files and you don't have them anymore. That's probably not a good situation you're in. Okay, what are FASTQ files? So FASTQ files are the files with the sequence data, and basically what they do is they have this suffix called .fasq, so .fasq.gz, and they store sequence data. So the reads of your sequencing, are in these FASTQ files. They are in a compressed format. Compressed means they are reduced in size, which is kind of similar like zip. So for example, when you have like a big PowerPoint presentation and you don't have enough disk space on your USB drive, you can use zip to make it smaller, and this will help you. And similar FASTQ files are compressed to make them smaller. Typically, the size of a FASTQ file is around from 2 gigabases to, well, let's say 100, 200 gigabases, and it gets really quick. You will spend a lot of your disk space in your directory. FASTQ files often come in pairs, and so that means your data set, for each data set, you have two FASTQ files, a file called R1 and a file called R2. And this is because today, when you sequence a library construct, you will sequence it twice. You will sequence it from the left side, and you will sequence it from the right side. Left typically is R1, right typically R2. Well, typically. There's also, years ago, or let's say 10 years ago, most of the sequencing was done in single-end sequencing, and then you will only have one read R1, which is from the left. Yeah. So once you have the files, what can you do with them? These FASTQ files need dedicated tools. So it means that you cannot open a FASTQ file in a text editor and have a look at the sequences. It would probably also doesn't tell you much. Just because of the size, you need tools that can process the data, and you will probably also need the computing power. Just a quick, I wanted to show you quick how the FASTQ format looks inside the files. So if you were to open such a file and you have a look at the first 4 lines, you can see that each read basically consists of 4 lines. So the first 4 lines are read 1. And the first line is the read name. The read name here looks a bit, well, there are a lot of numbers and symbols and so on, but actually it contains the sequencer, which is the A stuff here, so until here. The sequencing run, which is the 386. The flow cell code, so to say, which is this code here. And then it also contains the coordinates of the read, on which lane the read was done, the X and the Y axis, and also, I think, the tile. This is the name, and it contains this information. The next line would be the actual sequence. This is what you're interested in, what contains the data of your experiment, and which will be analyzed. Then the third line is typically a plus sign. This is just by convention. You won't see another character there. And finally, when we do sequencing, we have for each base, we have also a quality value, meaning how likely do we see the base at this position. These quality values are numeric, but to have a better compression of the data, to make the files more compact, they are encoded here as so-called characters. And each character stands for a certain quality value. So for example, an F could stand for a quality value of 40. Good. So now the question is, how do we know what is in our read data? And it often helps, in my experience, to first have a look at the library construct. So I would always suggest, if you have, for example, a certain single-cell sequencing technology, to go to the website, and there they have the protocols, but they also outline the final structure of the construct. And When you look at this, you can already guess what will be inside your reads. This here is a typical 10x library construct. You have a certain part called P5, which is the grafting part that attaches to the flow cell. You have a sequencing primer for read 1, which is called read 1 here. Then you have the 10x barcode, and Susanne told you all about, and the UMI. You have a poly-DT region, which is of the beat, but I will tell you later. And here we have the transcript. This is the information you're interested in. And finally, you have the sequencing primer for the second read, and barcode to identify the samples, and the P7 is another grafting part that attaches to the flow cell. Now, the read 1, when we look at this, it starts here, and it sequences the 16 base per 10x barcode and the 12 base per UMI. And this information is, of course, important, but then it will sequence the poly-DT region. And most of the time, you cannot use this information after position 29 for anything. So that's sometimes facilities, sometimes therefore only sequence the first 28 base pairs. Since it contains the barcode information for your single cells, it's often called the barcode read. Then we start with the second read, paired density sequencing, right? And we start sequencing here. Typically, we will sequence around 100 base pairs, sometimes only 50 or 150. This depends on your sequencing configuration or what you ask your facility to do so. And when you do this, you can see that you right away start sequencing transcript for 100 bases. Now, if this transcript part is short enough, you will also reach the poly-DT region. This poly-DT region, which, because we sequence the opposite strand, will show up as poly-A. But this is only the case if your transcript is short enough. Most of the time, you will not reach the poly-DT region. Yeah, only important for small fragments. Overall, this read just contains transcript information, and therefore it's called transcript read. We can confirm that this is indeed the case by just running a certain tool called FastQC. And FastQC is a tool for primary sequence quality control. And one of the plots it produces for you is a sequence composition plot. And this is shown here. On the x-axis, you see the position in the barcode read. So here's the start, here's the end. On the y-axis, you see the sequence composition in percentages, and the 4 lines are the sequence contents for T, C, A, and G. Just for your orientation, here is the sequencing construct, the P5 and the beat, and here's the transcript. So basically, this is the 3' end, and here's the 5' end. And when you look at this plot, you can see that there's a first part from base 1 to roughly 28, 29, for which we can see a balanced sequence composition. So all A, C, and T are present there, so to say. And this is, of course, understandable because the cell barcodes are random and the UMIs are random. So you would expect that you can see all bases at the same content, so to say. Then we see here this peak, and this is the poly-DT stretch of the beat or of the construct. And typically this will be around 30 bases. And this is still from the construct. And finally, we have like a third part, and because you have the poly-DT stretch and your transcript will attach with the poly-A like this, right? So, and therefore here the transcript region starts. So this is the 3' end, And the more you go in this direction, there's the 5' end. Good. We can do the same for the transcript read. And here we again have to remember that we sequence in the opposite direction. So that means now we have the 5' end of the transcript here and the 3' end of the transcript here. What we can see is that because this read contains transcript information and we sequence many different genes with different sequences, we can see that this transcript read has a balanced sequence composition. So it is already an indication that we have a good quality. The only interesting thing is this pattern here on the left. And this is like an unusual pattern. But remember, when we do the sequencing, our transcripts are like 1 KB, 2 KB, even 10 KB long, and the constructs will be the same. But the sequencer cannot sequence 10 KB. You have to cut down the reads or the fragments to a certain length of 600 bases. And therefore, we fragment the construct, and we use an enzyme called integumentase for this. Theoretically, this enzyme should cut completely unbiased. However, we often see that the integumentase has a certain sequence bias or preferred sequence distribution where it cuts. And that's why you see this strange pattern here. Okay, in general, I would advise to use FastQC as a first quality control because, it is easy to run. So you can run it on raw sequence data. It's just one tool you need to download, and you don't need any other tools. And you already get like an impression of your data. FastQC checks different sequence metrics, which are very basic. So like, for example, number of reads, the per base sequence content, the per base sequence quality, GC content, adapter content, and more. I have to say, however, when you run FastQC, it has these flags here on the side. So you see these green, red, and yellow buttons. And this is because FastQC automatically flags problems. So for example, here you would see that basic statistics here, so the first part in the report, have a green check mark and therefore are okay. But then the next one has like a red cross and they are not okay. The problem is the thresholds for deciding what is good and what is bad are for genomic data. So when you run data for FastQC on a whole genome sequencing library, these flags are good. You can use them. However, when you have single-cell RNA-seq or RNA-seq general and RNA-seq data in general, you cannot use them because it's different. So what you have to do is to go and check each matrix, have a look at each of the matrix. And here I would suggest to you some of these recommendations. What you should watch out is, first of all, an unexpected loss of reads. And I mean unexpected loss of reads. So let's say you sequence your target sequencing for 100 million reads. But after sequencing, only, let's say, 1 million reads is obtained by the sequencer. This is typically an indication that the library has already some problems because, well, typically in the sequencing facility, they are pretty good at targeting read amounts. You should also check for unusual sequence composition, and I've shown you how a typical 10x construct looks like. Also, you want to look out for undetermined bases, and this means bases that are not called. So typically, this will be n's, and this is problematic. So, for example, if you have in a cell barcode an n, well, then you cannot use the cell barcode, and it's also not good. And finally, also always look for adapter content. So now that you have done some very basic quality control and some checks, and we just discussed the format of the data, I will talk a bit about how, in general, reads are converted into counts and what pipelines there are. So before I start, I just want to outline you the basic steps of an bulk RNA-seq workflow, because the steps for a single-cell workflow are pretty similar, and you get an impression how the basic analysis steps for this data are. You start with reads, right, which are typically FASTQ files. Now the problem is that you do not know what these reads are, and of course you cannot blast them. But what you can do is you can do a process called mapping. And mapping means we take these reads and we compare them against a so-called reference. This is another sequence, typically longer. This sequence could be like a genome, it could be a transcriptome, it could be something else. Now, what you need to take care of here is that remember that transcripts, so RNA-seq reads, single-cell RNA-seq reads, bulk RNA-seq reads, are transcript. That means in many cases, the introns in the transcript are already spliced out. However, your reference, when it is a genome, it will still contain the introns. That means that your mapper has to be aware that there are certain parts it needs to splice out when doing the alignment. And therefore, it is called splice-aware mappers. Examples would be here, for example, star or top hat. Once you have done the mapping step, you have alignments. And alignments are typically stored in BAM files. Alignments contain the sequence information of the read, the position where it maps, and often they also contain how it's mapped. So for example, is there a gap in one of the alignments, or is it only a partial mapping? So, and these BAM files are typically the input for many other tools. And finally, of course, you want to get gene counts. And for this, now you know where the reads are, but in your reference, you don't know what the positions of the reference are mean. And therefore, you need gene annotations. And gene annotations usually come as GTF or GFF file. And I'll tell you which genes are in this position of the reference. And in this case, for example, I have two genes. I have a purple gene and a green gene. And all you need to do is then to run a so-called summarizing tool, which is just a tool that tells you for each of these regions or genes how many reads overlap to this region. And summarizing tools, there are typically one very well-known tool is feature counts, but there are others. In the end, you will end up with a table that contains the counts of reads for each gene. And then, of course, for each sample, if you run multiple samples. Now, coming back to the single-cell data, what are your options to do these steps for your single-cell data? The first option would be you are in luck, because it could be that a sequencing facility, a center, does it for you. This is nice because you don't have to take care of it and it doesn't take long. The drawback is that they might charge you for it and it's less flexible. Then for 10x data, there's the Cell Ranger pipeline, and this is provided by the company. And I will talk about this in more detail. I just wanted to tell that Cell Ranger, it offers lots of features. However, it can be computational intensive and quite slow. And therefore, there are some other pipelines available which are a bit, at least computationally, a bit better. And one of them is called Star. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And Star Solo is just an extension of Cell Ranger. And extension of the star mapper so that the mapper can handle single cell data. Star Solo is faster. The drawback here is you have to set it up for your data, and there are more options to configure and so on. And finally, another well-known tool for this data is Callisto. And Callisto is a very fast and very inexpensive RNA-seq mapper. Recently, there was also support for single-cell data that was enabled by the so-called bus tool scripts. And with Callisto, you're really fast. So Star Solo and Cell Ranger will, let's say, take an hour or two. And Callisto probably does it in like 20 minutes or something. The drawback here is that you need a really good annotation and you don't get all the features of, let's say, Cell Ranger. So now I will switch to Cell Ranger and tell you a bit about this pipeline. So what is Cell Ranger? And it is not one pipeline, but actually it is a set of pipelines. A set of pipelines offered by 10x Genomics. The main features of all these pipelines is that typically they convert reads to counts per cell and gene. They do some quality control for you. They provide options for visualization and exploration. And they also do some first basic single cell data analysis. Cell Ranger is open source, which is very nice because you don't have to have an account at 10x and you don't have to pay buy a kit or something. You can just download it and run it. It's Linux based, so you have to have some experience in Linux. But it is very easy to install. Actually, you just need to download it and unzip it. And the documentation of 10x is really good. So you will find a lot of details on their website, so I can only recommend it. Cell Ranger, I said that it's a set of pipelines. And I just want to show you what kind of Cell Rangers there are. The first one is actually Cell Ranger, so that's confusing, right? Because there's a set of pipelines called Cell Ranger, and the first one is called Cell Ranger. But it is actually the first 10x pipeline developed by Cell Ranger. And this is for transcriptome data, for surface antibody data, and CRISPR data. Then there's a pipeline, Cell Ranger VDJ, and this is used if you want to analyze T cell and B cell receptor data. So, for example, you can identify T cell and B cell receptor sequences per cell, which helps you in like immunity-related experiments. Another pipeline is called Cell Ranger Attack. And I guess the name already says it. It is for the analysis of chromatin accessibility data. Instead of genes, here you will analyze peaks. So instead of genes per cell, you will have peaks per cell. And finally, there's Cell Ranger ARC. And this is a special pipeline dedicated for multiome because there are kits from 10x which allow you to profile both transcriptome data as well as chromatin data together, so that you can analyze the gene expression profiles, and you also have an idea about the regulation of the genes. And for this, however, you need a dedicated pipeline called Cell Ranger ARC. So how does Cell Ranger work? And in the next slides, I will show you some of the details. I think that it is important to understand this because typically, let's say, for example, you start with counts, okay? Then, yeah. And if you understand the details, you understand the output. That's what I said. Okay, the first part is transcript read trimming. And I already told you that the transcript read contains adapter and poly-A sequences. And Cell Ranger, what it does is, the problem is here that these artifacts can be complicated. So for example, a poly-A sequence at a transcript, the mapper can also find the poly-A in the genome, which does not help. And here, Cell Ranger just trims the adapter sequences from the 5' end and poly A from the 3' end. And this information, it's not lost. It's also recorded in the output BEM file as certain called BEM tags, TS and PA. So when you have the results, you can for each read check what was trimmed. And then in next step, it will filter the cell barcodes, and this is because not all barcodes are really valid barcodes. And this is shown here. Let's say you have a set of observed barcodes that are in your reads, right? Here the barcodes are only 8 bases long, typically there's 16 bases. And CellRanger has a whitelist. So basically it has a list of barcodes that are allowed. You can get them, they are included into the software. And what it does is it takes this list and uses it filtering. So for example, when we check the first 3 reads, we can see that they match the whitelist, right? So we can include them in the further analysis. The same is for the next read, right? We can see that it is at the whitelist, and we add them as well to the output or to the reads that we process further. Now, what is if we have a read that has 1 base pair difference? Because here we have a read where the barcode just differs by the last base, but because of quality, sequence quality values, we know that it is a sequencing error. Therefore, we can say that we can correct this barcode and just include it here as the corrected version. And finally, the last 2 barcodes, we don't find them in the whitelist. We cannot correct them, so therefore these reads are discarded. So next, we come to the mapping, and now we will map the transcript read to the genome. CellRanger uses star for mapping so that we can do splice-aware alignments. And the mapping is done in 2 steps. The first step, the reads are mapped to the genome, and we do this. And then unmapped reads are discarded. Reads that map to multiple loci are discarded unless one of the loci is an exon. And finally, intergenic reads, reads that map between genes, are discarded. And if you set CellRanger up like this, intronic reads. The default is now CellRanger will include intronic reads. In the second step, the exonic and intronic reads are mapped to the transcriptome to make sure that they match annotations. And here we will discard antisense reads, which means reads that map on the opposite of the gene strand. And we also discard the reads that map to multiple overlapping genes. Once mapping is done, the transcript, the gene ID and name are recorded as BEM file tags so that you can later find out to which gene the read mapped. And all reads that pass the mapping step are used for counting. So now we have reads and we have alignments and we will do counting. The problem is read counts would be biased. So we have PCR amplification effects and we have low material and PCR will distort the counts. And therefore, in the 10x construct, you have UMIs. These are introduced before the PCR step and therefore are unbiased. That means that when you check a read, you can say that a read that has the same cell barcode, the same gene, and the same UMI originates from the same cDNA molecule, and therefore is counted as one UMI count. There's also done some sequencing error correction. So for example, if you have the same cell barcode and same gene, but the UMI differs by 1 base pair, you correct it. And uncorrected and corrected UMIs are recorded as BEM tags. So finally, we come to cell barcoding. And because we have now lots of cell barcodes, but we still don't know which cell barcode are good and which are not good. And for this, we use this cell barcode rank plot, which here shows the barcodes sorted by decreasing frequency. So here's the top barcode, here's the fewest barcode, and here the UMI counts. And we have like a graph, and we can divide this graph in multiple parts. The first part is the part with the large cells, shown here. And there's a glyph which typically indicates the number of cells. The second part contains small cells and ambient RNA. This is this glyph itself, and we don't know whether it is small cells or free-floating RNA. Then there's another plateau here, which indicates the number of gems or beads that were used in the experiment, because the lab, they will use 100K gems for the single cell experiment. So in the end, in this plot, you should see 100K again here. And finally, there's a part with the sequencing errors shown here. Cell calling is done in two algorithms. So first of all, there is a so-called ORDMAC algorithm, which calls the cells here, which are the high content RNA cells. Then the question is, what about this part here in the middle? And what the cell ranger does is the so-called empty drops algorithm, which builds like a gene profile of the ambient RNA shown here, right? It will then also build profiles for each barcode in this region here in between. And then it compares the profiles, and it just finds out which barcode is likely ambient, which barcode is not. So, and finally, I will come to the last part of the cell ranger, secondary and single cell analysis, and there the next talk will take over. So Cell Ranger already reduces the data, already does some single cell analysis. And the first step is usually to reduce the high dimensional data. This is done via PCA, and we focus on the first 10 components. Then this reduced data is used for clustering, and clustering here Cell Ranger does graph-based methods and also k-means methods. Then 2D visualization is done with t-SNE and UMAP. It will also identify for you markers for clusters using a negative binomial test. And finally, the results are exported as loop files. And 10x has a visualization tool for loop files with which you can already explore your data, which is quite nice because you don't necessarily need bioinformatics experience. You can use the loop file and already dig into your data. And further details will be presented in the upcoming talk.